Hi everybody, my name is Alex Brooker, and in this video, we'll be covering a key structure in muscle physiology, the neuromuscular junction, or the NMJ. It's a specialized synapse that serves as the critical link between the nervous and muscular systems, transforming cognitive output into action. Together, we'll break down the anatomy of the NMJ, its step-by-step mechanism of action, and its essential role in human physiology and motor control. Before we examine the neuromuscular junction in greater detail, it's important to understand the big picture. What is the neuromuscular junction? What does it do? Why is it important in muscle physiology? To answer these questions, we first need to understand the synapse. A synapse is the site where electrical or chemical signals are transmitted between two cells, either two neurons, or in this case, a neuron and a muscle cell. The neuromuscular junction is a specialized synaptic connection between a motor neuron and a muscle fiber. It functions to transmit an electrical signal, an action potential, from motor neurons to the muscle, enabling muscle contraction. The neuromuscular junction is essential in muscle physiology because it enables voluntary and involuntary muscle control, ranging from large gross motor movements like jumping to precise fine motor actions like writing your name. When the brain issues a motor command, it generates an electrical signal, an action potential, that travels down the spinal cord, through a motor neuron, and reaches the neuromuscular junction. There, chemical messengers, called neurotransmitters, cross the synaptic cleft and bind to receptors on the muscle fiber, triggering contraction. The neuromuscular junction relies on nerves, electrical signals, neurotransmitters, enzymes, ion channels, and molecular transporters to facilitate this process and prepare for subsequent motor commands. This highly specialized interface ensures efficient communication between the brain and body, coordinating cognition with muscular action. To understand the neuromuscular junction in greater detail, let's begin by looking at the core components involved. First, let's review the structure of a neuron. A neuron is a specialized cell that transmits electrical signals throughout the nervous system. There are three major neuron types: sensory neurons that transmit sensory information to the brain, motor neurons, which carry instruction from the brain to the muscles, and interneurons, which connect neurons within the central nervous system and facilitate pathways between sensory and motor neurons. A typical neuron has several key components: dendrites, a cell body, an axon, and a nerve terminal. Dendrites are branch-like extensions that receive signals from other neurons, called presynaptic neurons, and transmit them to the cell body. The cell body integrates these incoming signals and generates action potentials if the input is strong enough. The axon is a long, slender projection that carries action potentials away from the cell body toward other cells like muscles, glands, or other neurons. The nerve terminal, sometimes called the axon terminal, is a specialized region at the end of the axon. When an action potential reaches this region, it triggers the release of neurotransmitters, which cross the neuromuscular junction and bind to receptors on the muscle fiber, initiating muscle contraction. Now, let's look at the structure of the neuromuscular junction and how it enables muscular contraction. There are three main structural regions of the neuromuscular junction: the presynaptic region, or the nerve terminal, the postsynaptic region, or the motor end plate, and the area between the nerve terminal and motor end plate, or the synaptic cleft. The motor end plate is a thick portion of the muscle plasma membrane, or sarcolemma. To increase surface area, it contains junctional folds, deep invaginations that house nicotinic acetylcholine receptors and ligand-gated ion channels. Nerve terminal endings align with these folds, positioning neurotransmitter release directly adjacent to receptors to ensure efficient signal transmission. The synaptic cleft is the extracellular space between the nerve terminal and the motor end plate, where acetylcholine is released and diffuses before binding to receptors in the junctional folds. In the nerve terminal, the neurotransmitter acetylcholine, or ACH, is synthesized from acetyl-CoA, a product of mitochondrial metabolism, and choline, an essential nutrient, by the enzyme choline acetyltransferase. Acetylcholine is then transported into synaptic vesicles by VACHT, vesicular acetylcholine transporter. When an action potential reaches the nerve terminal, it opens voltage-gated calcium channels, allowing calcium to enter the cytosol. Calcium then binds to the membrane protein synaptotagmin, which acts as a sensor and triggers a protein assembly called the snare complex. This interaction allows the acetylcholine-filled vesicles to fuse with the presynaptic membrane, leading to the exocytosis of acetylcholine into the synaptic cleft. In the synaptic cleft, acetylcholine then diffuses to the motor end plate and binds to nicotinic acetylcholine receptors within the junctional folds of the sarcolemma. These receptors are ligand-gated sodium channels that open in response to acetylcholine, allowing sodium to enter the muscle fiber. As positively charged sodium ions flow into the muscle cell, and some positively charged potassium ions flow out, it results in a net influx of positive charge. This localized depolarization of the membrane is referred to as an end plate potential. If the end plate potential rises from the resting value of -100 millivolts to the threshold value of around -60 millivolts, it will activate voltage-gated sodium channels in the surrounding sarcolemma, triggering an action potential in the muscle fiber that propagates along its entire length and initiates muscle contraction. After acetylcholine is released and binds to the receptors of the motor end plate, the signal must be rapidly terminated to prevent sustained depolarization and muscle contraction. This is accomplished by the enzyme acetylcholinesterase, which is embedded in the synaptic cleft. It rapidly hydrolyzes the acetylcholine into choline and acetate. Choline is then actively transported back to the presynaptic nerve terminal by choline transporters. As discussed previously, choline combines with acetyl-CoA to synthesize acetylcholine, catalyzed by the enzyme choline acetyltransferase. Acetylcholine is then transported and packaged into synaptic vesicles by the vesicular acetylcholine transporter. This highly efficient recycling system ensures the neuron maintains a steady supply of neurotransmitters for the next action potential. Without this tightly regulated process, signaling at the neuromuscular junction would be delayed, uncontrolled, or bioenergetically unstable, compromising muscle coordination and motor control. To review, a motor command from the brain generates an action potential that travels down a neuron to the neuromuscular junction. At the nerve terminal, this electrical signal triggers calcium influx and the release of acetylcholine into the synaptic cleft in conjunction with the snare complex. Acetylcholine then binds to receptors on the motor end plate, generating an end plate potential that, if it surpasses the threshold of -60 millivolts, activates voltage-gated sodium channels and triggers a muscle action potential, ultimately leading to muscle contraction. The signal is rapidly terminated by the breakdown of acetylcholine via acetylcholinesterase, and choline is recycled back into the presynaptic terminal to synthesize acetylcholine. Together, these steps ensure the neuromuscular junction remains a fast, precise, and energy-efficient interface between the nervous and muscular systems to enable controlled movement. Through this explanation of the neuromuscular junction, we've broken down not only its anatomy, but the detailed physiological mechanisms that allow it to convert electrical signals into mechanical action. From acetylcholine synthesis and calcium-triggered vesicle release to receptor activation, ion flow, and muscle fiber depolarization, we've seen how each molecular step contributes to the larger goal of controlled and coordinated movement. This reflects both the precision and teleological purpose of the neuromuscular junction, to enable rapid, repeatable communication between the brain and skeletal muscle. By understanding its physiological underpinnings, we can better appreciate the NMJ's critical role in motor control and the highly integrated systems that make movement possible.